Assalamu alaikum dear students. Welcome to Dr. Roomi lectures. Today we are going to study the properties of sensory receptors. In our previous session, sensory receptors part 1, we have studied some of the properties of sensory receptors, and today we will revise some of those properties which we have already studied, and we'll study some more properties in detail. Receptors are sensory transducers, which means that when a stimulus is given to a receptor, it converts the stimulus energy into electrical energy. And this property is called as sensory transduction. All the receptors are biological transducers, and this property is called as transduction. When light falls in our retina, rods and cones convert this light energy into membrane potential change, and this process is called as phototransduction. When sound energy enters in our internal ear and that converts into electrical change, we call it auditory transduction. So this transduction in different receptors is called differently, like phototransduction, auditory transduction. However, this change in the membrane potential, which is also called as receptor potential or generator potential, it may be depolarizing, which means it moves towards positivity, or it may be hyperpolarizing. In most of the receptors, this is depolarizing change in the receptor potential, except in one sensation, which is visual sensation, where this change in membrane potential is hyperpolarizing, or it makes the membrane more negative. Receptors are specific to detect a specific stimulus. For example, touch or the mechanical stimulus is detected by the touch receptors or mechanoreceptors in our skin. So touch is adequate stimulus for our mechanoreceptors because the minimum stimulus energy of the touch can stimulate these touch receptors. And for our rods and cones in the retina, light is the adequate stimulus because the minimum light energy is able to stimulate our rods and cones. Touch is not an adequate stimulus for our rods and cones, and light is not an adequate stimulus for our touch receptors. So this property of receptors is called as specificity or differential sensitivity. A certain part in our skin or in some other region of body has receptors which belong to a nerve fiber, and that region is called as receptive field. For example, so some small area in our hand may have receptors for one nerve fiber, and that is called as receptive field for that nerve fiber. We'll study this topic of receptive field and its role in determining the acuity of sensations in detail. Muller's law or Muller's doctrine of specific nerve energies states that one nerve fiber carries a certain sensation, and the sensation carried by that nerve fiber is the one which is produced when the receptor is stimulated by its adequate stimulus. For example, the rods and cones in our retina, they are stimulated by the light, which is adequate stimulus for them. So the signal carried will be the visual signals or light signals. So even if you stimulate your rods and cones with intense pressure, like ball hits on the eye, once again the signals carried from our rods or cone will be the same, which is the light signal. So this is called as Muller's law or law of specific nerve energies. According to the labeled line principle, receptors detect a sensation and that travels along a series of nerve fibers. Like in dorsal column pathway, there is first, second, third, and fourth order neurons. Similarly, in the spinal thalamic pathways, and those neurons they take their signal to a specific part in our cerebral cortex, like in the primary sensory cortex. Labeled line principle states that the signal is detected from a certain part of our body, and then it travels along a series of nerve fibers in a certain pathway, and then goes to project in a certain destination which is in the cerebral cortex. This is called as law of projection. So the destination for visual signals is in our primary visual area, which is in the which is area 17 in the occipital lobe. The destination for our auditory signals or hearing signals is in the temporal lobe. Destination for the somatosensory signals like pain, touch, pressure, vibration from our skin in our hands and feet is in the primary sensory cortex in the parietal lobe. So this law of projection and label line principle are very important to understand a phenomena which is called as phantom limb pain. When the leg of someone is amputated, what happens? The person perceives pain as if it is originating in the missing foot, which means that when the nerve endings which are carrying the proprioceptive signals and the pain signals, when they are stimulated here in the cut stump, the signals of pain and proprioception which are coming from the foot region, they are perceived in the brain, and brain perceives as if the signals are coming from the foot. It's because wherever we stimulate those nerve fibers which are carrying signals from the foot, the signals will be perceived as if they are coming from the foot. So this phantom limb phenomena, it is helpful for us to understand the labor line principle and the law of projection. Weber-Fechner law describes the relationship between change in the intensity of stimulus and the change in the perception of sensation. We will use this Weber-Fechner law to describe why we are able to perceive a wide range of stimulus. Like we can hear the very feeble sound of a whisper, and similarly, we can hear a very loud noise. And similarly, our eyes can see a star at night and can also see bright sun during the day. So this change in the intensity of a stimulus can be perceived by our receptors, and we will use Weber-Fechner law to describe it. We'll study this law in a bit more detail shortly. Receptors can also adapt to the sensory stimulus, which means that when a stimulus of constant strength is applied on the receptor and is maintained, initially there is increase in the firing rate from the nerve fibers, but with the maintained stimulus, the firing rate or the number of action potential declines. So this rate of decline of the firing from the nerve fibers, if it is very rapid, then such receptors are called as rapidly adapting sensory receptors. If this decline in the number of action potentials with a maintained stimulus is slowly, then we call these receptors as slowly adapting receptors. We have already studied this topic of rapidly versus slowly adapting receptors in detail in a previous session, in which I have given an easy mnemonic to remember the names of rapidly adapting receptors. And the mnemonic is Miss Pacific wears a crown. Here Miss is for Meissner's corpuscle, Pacific is for Pacinian corpuscle, crown is worn on the head. So crown is for hair follicle receptors. So these three receptors are rapidly adapting receptors, and most of the other receptors like Merkel's disc, Ruffney's and organs, chemoreceptors for maintaining hydrogen carbon dioxide and oxygen level in our blood, the baroreceptors in our aorta and carotid sinus, they are all examples of slowly adapting receptors. Now we will discuss three properties of receptors in detail, which are receptor potential, receptive fields, and Weber-Fechner law. We'll talk about receptor potential in a little bit more detail, also called as generator potential. This is the membrane potential change which is produced when a stimulus is given to a receptor. Let's suppose this is a Pacinian corpuscle, one of the most studied mechanoreceptors. When a stimulus is given to this Pacinian corpuscle, there are mechanically sensitive sodium channels which open. Sodium being positive moves inward. Normally inside is negative and outside is positive. When this sodium enters to mechanically sensitive sodium channels, inside becomes positive and outside becomes negative. Now this change in the membrane potential, which is movement from resting potential towards less negativity or more positivity, this is called as receptor potential. So here this is receptor potential, which is moving from the resting value gradually towards less negative or towards more positive value. This is also called as generator potential. Here we must not confuse that receptor potential is not true action potential. If the value of receptor potential is less than the threshold, then action potential is not produced. But when the value of receptor potential goes beyond threshold, it becomes more than threshold, then action potential is produced. And less is the value of receptor potential, less will be the number of action potential produced, and more is the value of receptor potential, more will be the frequency of action potentials. So in case of depolarizing receptor potential, the nerve fiber is stimulated, or there are more number of action potentials which travel along the nerve fiber. But in some sensations, the receptor potential is hyperpolarizing or inhibitory potential. For example, in case of rods and cones, this receptor potential does not move towards positivity, it moves towards more negativity, which is called as hyperpolarizing receptor potential. And in case of hyperpolarization, it will be inhibiting the membrane. In most of the receptors, this potential is depolarizing receptor potential. This receptor potential is a graded potential and is different from action potential, just like end plate potential is different, just like excitatory post synaptic potential is different. So you should know the differences between the features of receptor potential and action potential. Receptor potential is graded, which means it can be less, a little bit more, or even more. But action potential does not have any grade. Action potential comes only in one grade. Either action potential happens or it does not happen, which means action potential obeys all or none principle, but the receptor potential does not obey all or none principle. It can come in different grades. It can be smaller, medium, or large. Secondly, the receptor potential can be summated, which means two smaller receptor potentials, they can be added up to give you a bigger value. But action potentials, they cannot be summated. Action potential has a refractory period, which means if one action potential is already there, you cannot produce a second action potential unless first one is over. But the receptor potential, it does not have any refractory period. The receptor potential, it is decremental with distance, which means if receptor potential is produced at one point, then along the nerve fiber it will decrease, but action potential does not decrease. Action potential, when it is produced, it will travel with the same magnitude along the nerve fiber. For example, here in the nerve fiber of the Pacinian corpuscle, you can see here, when this receptor potential, it stimulates the voltage gated sodium channels at this node of Ranvier, then action potential is produced. Action potential travels in a saltatory way along whole the nerve fiber with the same magnitude. So these are some differences between the receptor potential and action potential. So in most of the receptors in our body, this receptor potential is produced by change in the permeability of some ion channels. For example, in the tongue, when chemical signals are applied to the taste buds, there is opening of some kind of ion channels. Similarly, in our hair cells in the internal ear, the mechanical stimulus of sound, it causes the opening of or closure of the hair cells. Similarly, the mechanical stimulus in our Pacinian corpuscle and some other mechanosensitive receptors also opens ion channels. So in most of the receptors in our body, the stimulus, it opens or closes some ion channels, and in this way it affects the membrane permeability and thus affects the receptor potential, and that produces action potential along the nerve fibers. Another important property of receptors describes a relationship between the change in the strength of stimulus and the actual perception of that sensation. And the graph is shown here, and this is called as Weber-Fechner's law. When there is change in the strength of stimulus, initially there is greater change in the perception of that sensation, but with further increase in the strength of stimulus, what you can see here, this graph gradually flattens, which means that there is less change in the perception of that sensation. And this relationship is actually log relationship, and we can say that the perception of change in actual sensation is proportional to the log of change in the strength of stimulus. So this is a compressed scale, which means that our receptors have the ability to detect a wider range of change in the stimulus strength. For example, our ears can hear a whisper and also a louder noise. Our eyes can see a star at dark night and also a very bright sun during the day. So this is possible because of this property of receptors, that they can detect a wider range of change in the strength of a stimulus by a compressed scale, which is a log scale, and the law is called as Weber-Fechner's law. And in this way we can perceive a big range of change in the strength of stimulus. So this Weber-Fechner law, it shows the relationship between change in strength of stimulus and the perception of change in sensation. And the brain perceives the intensity of stimulus by increase in number of action potentials which are traveling to the brain, and also increase in the nerve fibers which are being stimulated. So brain detects How much is the strength of stimulus? Number one, by detecting the increase in the firing rate of an individual nerve fiber, and also how many nerve fibers are firing at a time. And in some parts of our body or in some receptor areas, there are some special cells which are stimulated to inform our brain about the intense stimulus. For example, in our hands, the lighter stimulus stimulates the mechanoreceptors, but the more intense damaging stimulus also stimulates the nociceptors or pain receptors, so which informs our brain that the stimulus is intense. In our internal ear, when there is a feeble and normal sound, only the inner hair cells are stimulated. But when there is a louder noise, the outer hair cells of the cochlea, they are also stimulated, informing the brain that the sound has become louder. So these are some ways by which the strength of stimulus or increase in the intensity of stimulus is informed to the brain. The next very important property of receptors is their receptive fields, which is an area on the skin from which one sensory nerve fiber picks up the sensory signal, or the area on the skin which, when stimulated, will cause production of action potentials in the concerned neuron. This is a first order neuron, and here this is the area in the skin, for example, from where this neuron picks up its signals. It means the receptors for this neurons are located here, and this much is the receptive field for this neuron. And here, these are the peripheral nerve endings of another first order neuron. These are not multiple neurons. These are the peripheral nerve endings of one sensory neuron. And here, this is the total area from where the sensory signals are picked up from the skin. And here, this is a larger receptive field. This is a smaller receptive field, which means that the nerve endings of one sensory neurons, they are more densely located in this smaller area. But here in this larger area, the endings of one sensory neuron or its receptors, they are less densely located. So it's a larger receptive field, it's a smaller receptive field. So it means if a receptive field is smaller, the receptors will be more densely present, and if a receptive field is larger, the receptors will be less abundantly present, or there will be less density of receptors. The receptive fields may overlap on each other, and they can also interdigitate. Those areas of our body which have smaller receptive fields, they are better able to detect sharp or acute sensation. For example, the receptive field for Meissner's corpuscle in our hands is a very small area which is in millimeters, and that is very important for detecting two-point tactile discrimination. But the receptive field for two-point discrimination in our calf is much bigger. The threshold for two-point tactile discrimination in our calf is like about 50 millimeters, but in our fingertips it's like 2 millimeters. And this is the reason why the touch sensation in our fingers and hands, that is more acutely localized, or there is better ability to discriminate the touch of two separately placed points in our fingers as compared to in our back or in our calf. This is also because the representation of those areas which have more receptive density or which are having smaller receptive fields is much bigger localization in our cerebral cortex. We have already studied sensory homunculus. You know very well that in sensory homunculus, the size which is given to our hands, our fingers, our thumb, tongue and lips is much bigger as compared to the other parts of our body, which means that these parts, they have smaller receptive fields, more receptive density. That's why they're more acute in discrimination of the touch sensations. Also in our vision, the discrimination of light signals coming from two separately placed points from a distance is much better discriminated. So the visual acuity and similarly the two-point tactile discrimination in our fingers, these are much better developed. It's because they are having smaller receptive fields. The bigger is the receptive field, less acute the sensation will be localized. Another important property of the receptive fields through which localization is further enhanced is that when a certain receptive field, for example in our hand, is stimulated, the center is stimulated, but the periphery of that receptive field, that is inhibited. You can see here from this much area the excitatory signals are going along the first order neurons to the second order neuron, but here on the periphery of this receptive field, on the edges, there are inhibitory signals which are going to the second order neuron. This is responsible for better localization of this tactile sensation. So we can say that the sensations can be better localized because of smaller receptive fields, which means there is more receptive density there. There is higher receptive density. That means better localization or more acutely perceived sensation. And also when the center of the receptive field is stimulated but the borders or the periphery is inhibited, then there is better localization of the area or the borders. So this happens in our VN and also in case of our hands, where 2-point tactile discrimination in hands and the visual acuity is much developed because of these properties. In this localization, our brain also plays our role. As I have mentioned, those parts which have better receptive density, they are bigger representation in the sensory cortex, as is for hands, lips and tongue and face. So this is all about the properties of sensory receptors. I would suggest you go through all of my lectures on sensory receptors, which means the classification of sensory receptors, sensory receptor part 1, this lecture on the properties of sensory receptors, then rapidly adapting and slowly adapting receptors, and mechanoreceptors. In some of these lectures, I have also given very important mnemonics to memorize the features and examples of sensory receptors. I hope you must have enjoyed this topic of sensory receptors. Thank you so much for watching this video. See you next time with another video.